Immunotherapy. When our body is infected by virus or bacteria, these external origin antigens can generate immune response. How about tumors? Tumors are originated from internal origin. Then in this case, how does the immune system can recognize this tumor, which were originally same with our own body? Important component for this activity is a major histocompatibility complex, so-called MHC. Our cells can present this MHC, and it can present epitope. Epitope is an antigen-like small fragment, and this can be recognized by our immune cells. For example, in T cell, there are T cell receptor can recognize those epitope only when it is presented by MHC. If the antigen or epitope is not presented by MHC, the immune cells does not recognize. Also, CD8 protein can bind to MHC and make sure that this antigen presentation is done by our own cell. That's why MHC allows immune cells to identify normal or abnormal cells. In normal cells, the proteins can be degraded by proteasome, and those fragments can enter into ER lumen, and some of those fragments can be bound to MHC and presented on cell surface. This fragment is just a normal protein fragment, and this is not recognized by immune system because this is part of our self structures. In tumor cells, the malignant protein can be generated and break down, and then if it is presented on cell surface, this abnormal antigen is recognized by immune system as a non-self or abnormal antigen. When cancer cell present the antigen on the surface, this antigen is called as a neoantigen. And that antigen should be presented by complex with the MHC. If that MHC is complex type 1, then this is interacting with the CD8 cell. CD8 cell is a cytotoxic T cells. Once this recognition is confirmed, then TCR, T cell receptor, recognize those neoantigen. By recognizing this strange new antigen, now cytotoxic T cell initiate the attack on cancer cells directly. In MHC class 2, it's not directly presented by cancer cell, it's indirectly by antigen presenting cells. For example, cancer cell can be swallowed by macrophage through phagocytosis, and the fragmented the antigen is presented by MHC2 class. Then in this case, the binding partner is a CD4, and CD4 cell is a helper T cells. And T cell receptor recognize those antigen, then activated helper T cell can engage other immune cells, such as natural killer, B cell, cytotoxic T cells, etc. Tumor cells can generate a very special environment, so-called tumor microenvironment. When tumor initiating cells start to grow, it forms some core, and then generate a differentiated, big chunk of a differentiated tumor. And they recruit new blood vessel generation, and macrophage, myeloid-derived suppressor cells, and dendritic cell, or NK cell, T cell, and B cells. Finally, cancer-associated fibroblast. So these kind of cells get together and forms very special environment for cancer. In tumor microenvironment, even though there are various immune cells, but they carry very strange character. In tumor microenvironment, the regular immune response is suppressed. So immunosuppression is observed. There are several mechanisms for this immunosuppression. One case is conversion of the immune cell's character into immunosuppressive cells. Normal immune cells, when they get activated, they should attack bacteria or tumor. But in these changed immune cells, they have seemingly helped the tumor and also suppress the immune response. And they can inactivate other immune cells. And TME can secrete other immunosuppressive factors. And eventually, the incoming immune cell number is also can be suppressed. The chemical drugs, like a chemotherapy or targeted the cancer therapy reagent, they can directly attack cancer and suppress those growths. But in immunotherapy, it engages our own immune system and reboost those immune system to attack the cancer tissues. So this is a concept of immunotherapy.